Okay, the next topic is anatomical quantification and personalizable compositional models. And I will tell you about the variability of spinal cord topology. And next, based on pictures, sacral 1 and sacral 2, lumbar 1 until 5, and low thoracic 12, dorsal root project to spinal segment containing motor neurons innervating legs and lower trunk muscle. Based on part 1, the figure 1a, and then therefore we aim to identify an arrangement of electrodes that would be suitable to target all of these routes across the human population. We first determine whether the individual variability of spinal cord anatomy was compatible with this aim. Neuroanatomical quantification of 27 spinal cord exposed a pronounced variability of spinal segment length, in particular across upper lumbar segment. That is, can you show in figure 1b, and then we complemented this survey with an analysis of the three-dimensional topology of dorsal routes because we showed that this topology determined the distribution of EES-induced electric potential along the routes. To enable this visualization, we optimized structural magnetic resonance imaging, or MRI, sequences that increase the three-dimensional spatial resolution while enhancing the contrast resolution between the cerebrospinal fluid and spinal cord tissues. Based on picture part C in figure number 1, and then we know that extended data figure number 1. Okay, the next slide is we talk about this contrast and about the semi-automated conversion of MRI and computed tomography or CT image into realistic anatomical models of the entire spine. You can show it in the figure number 1D. This visualization confirms the pronounced variability in the rostocauda distribution of dorsal roots related on the previous picture in 1b, and then instead we found that the weight of the dorsal roots in three zones were remarkably consistent, and this is related with 1b and extended data that my friend will continue about figure number 2. And then atas of spinal cord model, our pulse generator could accommodate 16 electrodes. Therefore, our challenge was to identify an arrangement of 16 electrodes that access the 16 target dorsal rods. Despite the variable topology of the spinal cord, we posit that resolving this challenge would require trade-off, and that identifying this trade-off will be contingent on computational models. We thus established a pipeline to generate highly realistic computational models of the interaction between EES and the spinal cord. This pipeline leveraged the verified anatomical and biophysical models of the Sim4Life compositional life science simulation platform to generate three-dimensional finite element models of the spine from CT and MRI scan. I'm so sorry, I mean MRI scan based on the figure number 1 and extended data figure 1. created model of root-led bundles, and their innervation patterns extended data. Video number 2 will continue explained more by my friend and functionalize these geometries with fiber models covering the entire population of efferent fibers. Figure 1F. And the basic and neuron electrophysiology solvers of the Sim4Life simulation platform then predicted the probability of frame that EEF preferentially requires large diameter efferents because nearly all A alpha fibers were recruited before the polarization of A beta fibers based on figure 1G. Based on the graph, you can show that what is A alpha or A beta. Then we use this pipeline to generate a freely available atlas of 15 personalized computational models that provide a resource to study the optimal arrangement of electrodes across a large human population. Okay, I think that's all about my part 1, and I think that will be continue with my friend, and it's time for you guys.